and for the next presentation here from Lund, we are joined by Cline Scientific and CEO Patrik Sundh. Welcome, Patrik.
Thank you very much, and good afternoon, everyone. My name is Patrik Sundh. I'm the CEO of Cline Scientific, and I will tell you a little bit about our company and some updates on milestones that we are we have reached and that we will have in the future. Since we have presented our company here at BioStock a number of times before, I will primarily today focus on one of our projects that we're trying to drive into the clinic called StemCART, and we feel that we have some exciting news and coming milestones for that project. But first, a little recap about Cline. We have a patented nanosurface technology, which we use to study and control different types of cells. We apply our technology into two internal R&D projects. One, CellRACE, that involves a cancer diagnostic product, and the other called StemCART, as I said, which involves stem cell therapy designed to repair cartilage damage. We also have, since the beginning of the year, a collaboration with AstraZeneca, where they are they're in the process of testing our technology for use in a stem cell application with a different focus than our own, but in a very similar way. So, therefore, I want you to know that Cline today is an R&D company with a strong focus on these two projects and to drive them into a clinical phase, nothing else. Our technology, again, I think that you've seen this before, nanoparticles that are arranged in a certain pattern on surfaces. We're capable of replicating naturally occurring gradients that are the ones responsible for different types of cellular responses in the body. And we can also design these surfaces in many different ways. Therefore, they have a multitude of uses and serve as our platform technology. Our company has a history. We went public in 2015 on the Spotlight Market, and we moved our company to NASDAQ First North Growth Market in the beginning of the summer of this year. Our business model is we're a preclinical stage R&D company, as I said. We focus on building projects with high commercial value, and the revenue stream should come from either out-licensing these projects or divesting them in the future, and I will tell you about when. The two projects, as I've mentioned, CellRACE is a diagnostic project, and StemCART then is a stem cell therapy for damaged cartilage repair. I will very briefly dive into CellRACE, the diagnostic product. It's an IVD product for the early detection and determination of the metastatic potential of a breast tumor. It's a live cell-based assay looking at tumor cells, cultured tumor cells from the tumor over time. And eventually, we expect this product to be used for not only breast cancer, but other types of metastatic tumors and cancers as well. Let me see here. 90% of all cancer-related deaths are due to metastatic or the fact that cancer is spreading. This is today very difficult to detect before they already have spread, and therefore treatment is not optimal. The current methods focuses on different aspects, different biomarkers, genetic expressions, or the classical histopathology, whereas our assay is based on, as I said, live cells, therefore taking the entire reaction of the cells into account. What we do measure is, and what we focus on, is how the tumor cells can move. There are different types of movement. So far we've focused on directional migration, in other words, how far or how much, if you will, a tumor cell can move, which is an essential characteristic in order for tumor cells to spread out the body. They have to break out from the primary tumor and out into the bloodstream or possibly the lymph stream. This is something that is quite complex, and therefore we are developing an algorithm to be able to analyze all these movements that we are recording, and we will expand testing to a number of different cell lines that we can mix to make sure that we can differentiate between non-metastatic and metastatic cells in this project. So, StemCART. StemCART is meant to repair degraded or destroyed cartilage in different joints, notably knee joints, shoulder joints, and hip joints. Damaged cartilage is something that leads to chronic pain, and it severely limits and restricts people's lives. It's very difficult to live with. There is no cure, and the way to treat this is primarily by pain management, constant pain management. In some cases, there are some surgical procedures whereby you can operate and replace the joint with a prosthetic, plastic prosthetic. Problem there is that they don't last too long, and it also restricts what you can do or what you cannot do. There are some emergent treatments with mesenchymal stem cells, but it's not quite done yet. So currently there is no fully functional regenerative cartilage solution for these types of problems. What then is the StemCART approach? Well, what we have done is we have first taken cartilage cells and reprogrammed them back to what's called iPS cells, or as you see here, induced pluripotent stem cells. Then those cells, now stem cells, have been differentiated in a certain way on Cline's unique surface technology. And when we do that, we eventually get new chondrocytes or cartilage cells that are optimal to replace the damaged cartilage. And these cells are then, or will then, be integrated into a support matrix, which we have developed ourselves too. And this forms then an implant that can be implanted into the joint during a surgical procedure, a very simple surgical procedure. This will be a first-in-class on the market, and we also intend to make this the best-in-class in the market, and this has not been done before. The market is very large. I think these numbers are they are pretty moderate. It's estimated that up to 300 million people or patients suffer from serious joint damage, that is cartilage damage, at any given time. The sums of money spent on hip and knee replacements, as you can see, are enormous. And we think that with this type of a therapy, about 90% of the total market is addressable. Where are we then? Well, the roadmap for StemCART looks fairly much like any other clinical product. And you can see the top line, what it looks like in general. Below you see some boxes, what we need to do and what we have done. The green ones are accomplished already, are done. The yellow ones are what we're doing right now, and the red ones is what we will do. What is circled in the blue rectangle here are the steps that Cline will take. That's what we see as our job, so to speak. We need to partner to get this to the market with a larger company that can take it through phase 2, 3, and obviously market penetration or market to introduction. And that is something that we will do in the future. I want to point this out since some people told us that and speculated that we have a very long time before we can exit Cline's part in this. And it's not true, because our job is, as I said, to go through phase 1 maybe, but not more than that. A partner should be there to take it further after that. To get a little closer to this, where are we today? What are we doing? This is what we have done. We are in the middle of the proof of concept, which is a necessity when you're talking cell therapy. We will do an ex vivo test starting hopefully before Christmas this year. And the specific upcoming goals are to, number one, receive ethical permission to do some work on patient samples. We have sent that in already. We will then, right after we receive that, commence the ex vivo study that I talked about. We are currently or concurrently developing a method to measure the cell viability, how effective or how alive the cells are. That's obviously important. And we also have a project with Chalmers to combine the matrix that we already have developed with the cells and the cell functions. And we expect the initial results from our ex vivo tests sometime the first half of next year. The ex vivo testing will start, as I said, late in the fourth quarter of this year. It will be on human cartilage. We're doing this together with some surgeons that we're working with. And we will use cartilage that is removed from patients during surgery. And as I said, the ethical permit has been applied for, and we expect that to come through in the coming months. Benefits of this type of cell therapy is that, number one, that we can regenerate what's called articular cartilage for a large group of patients. This has been tried many times before, but not been successful in the sense that the cartilage does not last very long. We believe we have a solution to remedy that problem. And also, since our therapy is based on iPS cells, stem cells, it will be an allogenic cell source that allows the scale up of this cell therapy, which makes it financially justifiable, as opposed to the autologous stem cell therapies that are available today. In other words, you can only use your own stem cells from your own body. In addition to this, when we are successful with our own project, we believe that our technology and our methodology can be expanded to a number of cell therapy areas for other types of functional cells, such as what we have communicated in the beginning of the year. We are, as I mentioned, in collaboration with AstraZeneca, who is trying our technology within a project, an EU-funded project called VA-Cure, where they are trying to understand the mechanisms of what's called vascular anomalies. That means defects in your blood vessels. They are trying to differentiate also iPS cells, but into a different type of functional cell, epithelial cells, that is then hopefully going to be used in different types of therapeutic or different therapeutic ways to treat these vascular anomalies. Vascular anomalies are very prevalent today in the total population of the world. The complications can be very benign, birthmarks, but it can also be lethal if you have it in the brain, for instance, and a blood vessel will break. Many of these conditions cannot be treated, and the ones that can, they need lifelong medical management. In other words, there is no cure, and for these things, a cell therapy obviously would be a cure. So, some key takeaways from Cline Scientific today. We are very involved in developing new technologies for the life sciences. We have 2 clinical projects or preclinical projects that are on their way into the clinical phases with high value potential, both of them. And we think that you should look out for the upcoming results of our ex vivo testing sometime next year. And also progress in our cancer diagnostic project, CellRACE, where the prediction algorithm that we are developing will be very exciting to see how that can be used. And we believe that will be a breakthrough for us. Well, with that, I will leave you and thank you for listening, and hope that you're excited about this as I am, and we'll see each other again in not too long.
Thank you so much for the presentation, Patrik. Just had a couple of questions for you.
Mm-hmm.
You talked here about the testing the StemCART project on human cartilage tissue, which you're expecting to start Q4 this year. What are your hopes of eventually bringing it to human clinical trials?
Those are to clinical trials. We are very hopeful, and we see that as obviously we need good results, just like any project needs. But after this ex vivo study, we have some other preparations that we need to do, and then we will apply for permission to start phase 1. So in our minds, a phase 1 study should be possible within a year to a year and a half to get started.
And if we look a little bit broader and look at the competitive landscape, how would you describe that landscape and Cline's position on it?
That's a good question. That has to be answered in 2 different ways. If you look at Cline's technology as such, is unique. There, as far as we know, no one is working with nanosurfaces to introduce different biomolecules to cells and thereby elicit or responses or control cells, as we say. So there is very little or no competition from that aspect. When you come to our different clinical projects, though, that's a different story. The StemCART project is something that, not the StemCART product, but to repair cartilage, has been tried before, and there are a number of projects in clinical phases. However, they do not use the type of stem cells that we do. They are generally not allogenic, and it's questionable if the quality, if you want to call it that, of the cartilage that is delivered will last for, let's say, 20, 30 years. As far as the cancer diagnostic project goes, we feel that will take the diagnosis or prediction of metastasis much further, but obviously there are tons of biomarkers, genetic expression, etc., etc. So there is competition, of course. It's a good thing to have competition, though, because that means we don't have to build that market ourselves. So it's not necessarily a bad thing.
No. And finally, then, you have been on the Nasdaq First North Growth Market for a few months now. What are your long-term goals of being listed on Nasdaq?
Well, the primary reason for being listed on that, or to move to Nasdaq, was the fact that when we approached international investors as well as the larger pharmaceutical and life science companies, of which only one, as you know, is Swedish, they don't. When you say that you're listed on Spotlight Market, they don't know what that is. So there is quite a bit of uncertainty in what type of a company is Cline when you want to go that path. There is nothing wrong with Spotlight Market, but for us, number one, absolutely have to or would like to look for international investors, as well as, obviously, as I've said, our goals are to find a big-time partner in the life sciences. Then you need to be, if you are a listed company, you need to be on a stock market that is recognized and well known, and that's the primary reason for us moving to Nasdaq. Nasdaq has the name; it's a bigger market, etc., etc. So that's obviously it gives us access to to a bigger market and more investors here in Sweden as well. But long term, it's actually the international aspect of it.
And with that, we thank you so much for coming.
Thank you very much.